My lab's focus is to identify and characterize tumor suppressors and oncogene in cancer. And contrary to Paul's research interest of generating technologies, I usually use different technology which is already available for us to use. So among the different projects that we have been doing was to identify tumor suppressors and characterize them. So I'm going to talk about two of these projects today. And the talk has been divided into two parts. In the part one, I am going to talk about identification of tumor suppressor genes using large-scale in vivo RNAi screen. And also I'll present a screen where we have characterized tumor suppressor gene, which we have identified from a previous study using a genome-wide RNAi screen. So coming to the first part, as most of you will be aware that these are the two classes of genes which are mostly deregulated in cancer: oncogenes, which are activated and promote cancer, and tumor suppressor genes, which prevents cancer in normal cells. But when you have loss of tumor suppressor genes, it actually leads to activation. It leads to transformation and other events. So this is a general paradigm for cancer, is that you have normal proto-oncogenes and tumor suppressor genes, which does not have abnormal functions, and then you have oncogenic amplification or mutations which will activate oncogenes. But unless the tumor suppressors are inactivated, these oncogenes will not be able to transform and cause cancer. So in a very general sense, the activation of oncogenes and loss of tumor suppressor genes is what is going to lead to cancer. So now, how to identify these genes? There have been several studies using genome-wide RNAi screens in hematological malignancies where they have used mouse system to do screens and identify tumor suppressor genes. But the question is how to do it in solid tumors, which is much more difficult to isolate cells and then reimplant in the mouse and then identify tumor suppressors. So we tried to do a proof of principle screen using a very simple system, which is the NIH-33 cells system, which is, these are immortalized cells but not transformed and can be rendered by a wide variety of oncogenic stress, oncogenic events. And it's been shown that these cells have been used to characterize newly identified oncogenes. So what we did is we took mouse NIH-33 cells, which are immortalized, as I said, and then we transfused these cells with mouse shRNA libraries. And this library comprises of 25,000 shRNAs, 25,000 genes, and targeted by 65,000 shRNA. So the way we packaged these libraries were 5,000 shRNAs per pools, and those pools were used to infect NIH-33 cells, and those pools then eventually injected in immunocompromised nude mice model, which will be able to accept xenograft as well as the cells which also can even elicit immune response. So after injecting these cells, we allowed the tumors to grow, and then isolated tumors from 7 out of 10 pools that we injected. So 7 out of 10 pools were able to cause transformation of the cells, and in total we identified 49 genes from this screen. And so first thing was to see how many of these genes are false positive and how many are the real positive. So first we started with a cell culture model of cellular transformation, which is soft agar assays. And as many of you will know, that soft agar assay is one of the ways to identify transformed cells. So non-transformed cells will not grow and won't form colonies, but when you have loss of function events, it will cause cellular transformation and it will allow cells to grow. So out of the 49 genes that we identified, 28 of these genes were able to cause encourage independent growth. And to confirm this in mouse, we took these genes and then injected these genes. So we individually knocked down these genes in NIH-33 cells and showed that loss of any of these genes lead to cellular transformation. And it's important to note that we were able to identify some very well-established tumor suppressor from this screen, like STK11, which is LKB1, CMA3B, which is another tumor suppressor, and IGF2R, which is one of the very frequently down-regulated tumor suppressor in colon cancer and several other cancer types. So now the next question was, if these genes are coming from NIH-33 cells, they are causing transformation, what kind of tumors they are actually associated with, if they are real tumor suppressor? And general understanding is that they are either lost by epigenetic silencing or they are lost by mutations. So what we did is we analyzed several different tumor types for these tumor suppressor and found that 24 out of 28 genes were down-regulated in human squamous cell carcinoma samples. So the criteria was to at least they were twofold downregulated in at least 70% of the samples that we have analyzed. Further, to understand the mechanism of these, the downregulation of these genes, we collaborated with an Australian group who had data on squamous cell carcinomas, both transcription as well as DNA methylation data. And with them, we have been able to identify at least 9 genes which were significantly promoter hypermethylated out of these 24. So at least 9 out of 28 candidate tumor suppressor genes are downregulated by promoter DNA methylation. And we also did some surrogate assays to show that these are actually squamous cell lung carcinomas tumor suppressors. So we took squamous cell lung carcinoma cell line, and we ectopically expressed these tumor suppressor. And these figures are colony formation assays. And when you ectopically express a cDNA, which is a tumor suppressor, it will prevent growth of these cells. And this has been used to characterize other tumor suppressors. So we find that many of these tumor suppressors that we identify were able to reduce proliferation in culture, and same results we were able to identify, we were able to achieve using a nude mice mouse model. So non-silencing or vector containing squamous cell lung carcinoma tumors grow fine, but when you ectopically express any of these cDNAs, it prevents the growth of these tumors in mouse. Next, to understand the mechanism of the action of these tumor suppressor, we performed different assays, and the two most common events that tumor suppressor actually execute in cells are induction of apoptosis or induction of cell cycle arrest. And senescence, which is a form of cell cycle arrest, is what our lab focuses on. So we tested those two aspects. So we find that several of these tumor suppressors, when you ectopically expressed, cause an increased caspase 3 cleavage, which is a marker of increased apoptosis. And we also find that some of these genes, when ectopically expressed, has increased senescence-associated beta-galactosidase A activity, indicating that some of these genes are actually causing cellular senescence. So to conclude this part, and I want to add one more thing, for which I am not showing the data, is the squamous cell lung carcinoma issue, that why these genes are so important for squamous cell lung carcinoma. So we recently found, and we are still working on it, is that squamous cell lung carcinomas has amplification of FGFR receptor. And we find that when you overexpress FGFR receptor in NIH-TD3 cells, you can cause transformation. And we find that so many of these genes that we identified are actually attenuating FGFR signaling. So essentially, it actually tells that that's how these genes are so well intricately related to squamous cell lung carcinoma. So to conclude this part, our mouse tumorigenesis screening strategy identified at least 16 genes that have all the expected properties of tumor suppressors, like it increased growth in soft agar and promotion of tumorigenesis in vivo following inactivation, frequent downregulation in human tumor samples, and inhibition of colony formation following ectopic expression and tumorigenesis in mouse. And this strategy that we have developed is broadly applicable. We are trying to do other screens also using other immortalized cells. This is another example of MCF10A T cells. These are HRAS expressing immortalized cells. These are not transformed, but when you introduce shRNA library and inject into the fat pad of mouse, it leads to tumor formation. So this is another example of how you can use immortalized cells system to do this kind of screens. And more importantly is that the approach that we are taking is complementary to cancer genome sequencing efforts, where they identify mutations, identify epigenetic changes, but they have not functionally characterizing what they have been identifying. So coming to the second part, since once you have a tumor suppressor, how do you really characterize a tumor suppressor? So I'm going to talk about a tumor suppressor which we have been interested for a very long time, and which is the insulin-like growth factor binding protein 7. And I'll show you how we have characterized this tumor suppressor, how it functions, and how it regulates tumor genesis in general. So this tumor suppressor was identified from an original screen which we have performed in 2007-2008, and to identify genes which are required for BRAF-mediated senescence and apoptosis. This screen was done in primary foreskin fibroblast and confirmed in melanocyte to show that we have identified 17 genes. When you knock down, it prevents BRAF-mediated senescence apoptosis. So these are primary foreskin fibroblast with non-silencing shRNAs. And when you infect these non-silencing shRNA with BRAF V600, it almost stops the growth of the cells, either by inducing large amount of apoptosis and some amount of senescence. And then also, when you knock down the genes that we identified from our screen, it prevents the BRAF's ability to cause senescence and apoptosis. So one of the genes that I'm going to talk about is AGVP7. We try to characterize it in in the paper that we published previously, and this was the model that we generated using that. So we found that in melanocyte, IGF-BP7 is normally expressed through a BRAF-Macaroc signaling pathway, and it has a controlled proliferation, because if you knock down IGF-BP7 in primary human melanocytes, it provides a slight growth advantage. Interestingly, but when you have mutation in BRAF600E pathway, it causes increased activation of Macaroc signaling and causes increased production of IGF-BP7. And in this context, IGF-BP7 activates senescence inducing genes and causes senescence of BRAF-Macaroc signaling. And it's very interesting to see that if you analyze NEVIs, most of the NEVIs analyzed has a reduced fast-work staining. And we found that in melanomas which are BRAF-V600 positive, we have inactivation of IGF-BP7, and although these cells were still sensitive to IGF-BP7 induced apoptosis. So you can supplement IGF-BP7 from outside to get a clinical outcome or treatment of melanoma. This work that we have done is actually been recently recapitulated in a very different model. It's very interesting to see. And this is a syndrome which was very uncharacterized in terms of what the genetics. So it's called familial retinal arterial macroaneurysm. So it causes retinal defects and it has heart-related problems. So this group has actually found that IGF-BP7 has a splicing event mutation in this disease, and this leads to increased IGF-BP7 expression, but the function is completely gone, and it leads to increased Macaroc signaling. So it's interesting. Maybe Marcus's model may also have some kind of phenotypes in retina or heart. So this is the only human model unrelated to cancer known in which BRAF-Macaroc signaling, increased Macaroc signaling lead to this. And they've also, similar to us, find that IGF-BP7 is a negative regulator of BRAF-Macaroc signaling, and loss of IGF-BP7 leads to this phenotype. And in addition to the whole BRAF-Macaroc signaling pathway, there is another reason we wanted to follow up this tumor suppressor. And IGF-BP7 all functions as a tumor suppressor in almost every single cancer type tested so far. So it's as broadly mutated or epigenetically silenced as P53. So it's very broad. So these are some examples which shows that it's downregulated in colon, it's downregulated in breast, it's downregulated in lung, it's downregulated in thyroid, and all of them it acts as a tumor suppressor. In addition to that, we have performed some analysis where we show that because it's a secreted protein, you can supplement it from outside. And when you supplement them in a mouse model, you can have therapeutic benefit. It's a lung colonization assay using a metastatic melanoma cell line. Also, we analyzed the NCI-60 panel of cell line and found that several different tumor types, specifically colon, to a certain extent lung, melanoma, breast, and to some extent ovarian cancer cell lines show sensitivity to IGF-BP7. But there were a broad range of cell lines which has no sensitivity. So this has led us to ask why some of the cell lines are sensitive and why some of them are not. So this we can only answer once we understand how IGF-BP7 acts as a tumor suppressor. So to do that, we set up a screen using a cell line which is SKML28. As you will see here, this is one of the cell lines which is very sensitive to IGF-BP7 induced apoptosis. So the reason to choose this SKML28 cell line was to get a minimum possible background in the RNAi screen. So similar to the previous screen, we transduced these cell lines with shRNA library pools. Here we used lentiviral shRNAs, then do puromycin selection, and then transfected with IGF-BP7 cDNA. And essentially, we are looking for events which will allow survival of the cells in presence of IGF-BP7. So you collect, and we don't have to worry about 100% transfection because it's secreted protein and it functions in a paracrine fashion, so it still kills even if the cells are not, it doesn't express IGF-BP7 cDNA. So the cell surviving colonies we isolated and then PCR cloned and identified and validated the candidates. This screen has allowed us to identify 21 21 genes which, when knocked down, prevents IGF-BP7's ability to induce apoptosis. So every single gene is essential for IGF-BP7 to induce apoptosis. So the first thing that we did to ask was, because it's a secreted protein, we wanted to see if the mechanism by which these genes are working is by preventing IGF-BP7 internalization, and that will explain why they may act as a tumor suppressor or why they act as a regulator of IGF-BP7 induced apoptosis. So we labeled IGF-BP7 using Alexa Fluor 568 and then added on SKML28 cells, and then incubated for 24 hours, washed the cells, and analyzed with confocal microscopy. Out of these 21 genes, we were able to identify two shRNAs which two genes which upon knockdown prevented IGBP7 internalization. I'm going to talk about one of them today is IGF2R. The reason IGF2R was interesting is because it acts as a receptor, and a receptor for IGBP7 was so far unknown. So this actually provides you a new opportunity to actually test a receptor of IGBP7. And IGF2R is a very well established tumor suppressor, and the mechanism of action of how IGF2R acts as a tumor suppressor is also little unknown. So this allowed us also to give provided us with an opportunity to characterize two unrelated tumor suppressors and bringing them together and try to characterize. So without going to much of the data that we have generated in last 1 year, I'm going to tell you about the two models that we tested to identify how IGF2R functions as a receptor and how IGF-BP7's ability to bind to IGF2R regulates its ability to induce apoptosis. So the first model that we tested was a very simplistic model, and the idea was that it simply binds to IGF2R, gets internalized, and after internalization it activates signaling or transcription regulation leading to apoptosis. This may still be true in the other model too. But the second model was a little more complex and brought other candidates that bind to other receptors. So we considered IGF-1R, which is a pro-survival signaling, and IGF-2R, which is anti-survival signaling. And a regulator of these two signaling is IGF-2. So IGF-2 is actually bound in a normal condition, binds to IGF-2R, which internalizes it and causes a lysosomal degradation of IGF-2 and leads to pro-apoptotic signaling. And IGF-2 binds to IGF-1R, leads to pro-survival signaling. So we are expecting in a normal scenario where there is no IGF-BP7, this will lead to cancer cell proliferation because there is a balance between survival signaling and apoptotic signaling. So what we try to do is we generate a synthetic medium with and without IGF-BP7 and different combinations of IGF-2R, and we showed that if you have IGF-BP7, IGF-BP7 actually binds to IGF-2R and increases the affinity of IGF-BP7 to the IGF-2R receptor and prevents its ability to bind to IGF-1. So it leads to reduced pro-survival signaling through IGF-1 receptor and increased lysosomal degradation of IGF-2 by pro-apoptotic signaling and eventually causes cell death. And so this is what we think is the mechanism. We are trying to do some experiments in mouse, but we are still having trouble generating the knockout for IGBP7 because there are several family members which have a very high similarity in the same regions where we are trying to generate the knockout. So the second very interesting thing that we found from this screen was this gene called ADR. ADR is a very well established RNA editing enzyme. And before going to the detail of how this functions to regulate IGBP7, I just want to give a brief overview of how RNA editing works. So this is a general central dogma of molecular biology that DNA is transcribed to RNA and RNA is transcribed to generate protein. And there are other processes actually in addition to this simple step. You have splicing, and after splicing you can still have modification of RNA that leads to editing. And this editing can generate different proteins, a wild-type protein and additive protein, and these proteins can be different in many ways. For example, this RNA editing, A2I RNA editing, which is the most common RNA editing, can alter the primary sequence of the RNA. So it can generate a stop codon, it can create a new amino acid, and it can also create splice site modulators. The second thing is A2I RNA editing that can alter the secondary structure of the RNA. So its ability to bind to other proteins or other RNA will also change. In our case, what we found is, first of all, we wanted to see is IGF-BP7 is direct target of RNA editing, or it's a indirect effect that we are seeing. So we came across a paper from George Church's group, which came out in Science, and he found IGF-BP7 as one of the editing targets, and he found two sites which were edited in human genome. So what we did is we took unedited cDNA and then generated mutants by generating the edited form or a mutant where we had replaced the edited form with alanine. So to our surprise, what we find is the wild-type protein has this 25 kilodalton band, which is very little in the amount. When you cause this editing defect, it causes increased proteolytic cleavage of this protein. However, when you create a alanine, the proteolytic cleavage goes up further. We have confirmed these the sites of cleavage using mass spectroscopic analysis, and to our initial analysis shows that it's a matriptase site that is created, and it's only the change in secondary structure which leads to increased proteolytic cleavage. Some of our studies show that this 25 kilodalton protein is more effective in binding to IGF-2R receptor. So it may explain why it's able to induce apoptosis more and why the RNA editing is required. So to test that directly, we did this experiment where we took the unedited IGF-BP7 cDNA and then edited IGF-BP7 cDNA, and then transfected in SKML28 cell line that either had a non-silencing shRNA or two different shRNAs against ADR. So as you can see, that unedited protein in presence of ADR shRNA cannot induce apoptosis. However, edited IGF-BP7, whether IDR is present or not, it can still induce apoptosis, indicating that RNA editing of IGF-BP7 is required for its ability to induce apoptosis. And this is proven further that if you take a purified recombinant protein and edit on the cells, it can induce apoptosis, because you don't require editing once the protein is made. So to conclude this part is our genome-wide RNAi screen has identified 21 genes, all of which are required for IGF-BP7 induced apoptosis. IGF-2R functions as a receptor for IGF-BP7, and IGF-BP7 editing affects ability to induce apoptosis. And finally, our experimental approach can be applied to identify genes that are required for growth inhibitory activities of tumor suppressor or pro-apoptotic activity. So it's a general approach, and these are the lab members without whom I could not have finished anything. And for these two projects, we have collaborated with Michael Green Newmas and Shale Dogra, who is a bioinformatics collaborator for us in Singapore. And also want to thank all my mentors and friends, and who has always been very encouraging to everything that I do, and the money, which is very important to do any research. Thank you very much.